Welcome everyone. Today I will discuss about bacterial structure. Bacteria is a prokaryotic cell. It has some essential components. And some non-essential components. Essential components are necessary for survival, and bacteria cannot survive in absence of any of the essential components. But non-essential component is not mandatory for survival, and bacteria can survive in absence of non-essential components. Essential components are cell wall, plasma membrane, ribosome, mesosome, nucleoid. Essential components are cell wall, plasma membrane or cytoplasmic membrane, ribosome, mesosome, nucleoid. And non-essential components are capsule, pili, flagella, spore, granules and glycocalyx. So these are essential components and non-essential components. Non-essential components are capsule, pili, flagella, spore, granules and glycocalyx. Now come to this figure. The outermost layer, this is capsule. Capsule is a gelatinous layer covering the whole bacteria. It is gelatinous layer that surrounds the entire bacteria. Chemical nature of capsule may be polysaccharide or polypeptide. Most of the bacteria has polysaccharide capsule. Like Neisseria gonorrhoeae, Haemophilus influenzae, Streptococcus pneumoniae, Klebsiella pneumoniae, Streptococcus pyogenes, etc. This bacteria has polysaccharide capsule, and in case of Bacillus species, we found this polypeptide capsule. This capsule has antiphagocytic property. That means capsule provide protection against phagocytosis, and this capsular polysaccharide antigen is used for vaccine production. Example of such vaccine is pneumococcal vaccine. The layer immediately below the capsule is cell wall. Cell wall provide rigid support to the bacteria and prevent osmotic lysis of bacteria. There are some differences between cell wall of gram-positive and gram-negative bacteria. I will discuss later with figure. And the layer inner to cell wall is cytoplasmic membrane. This cytoplasmic membrane helps in active transport of different molecules within the cell. Now this is plasmid. Plasmids are extra chromosomal, that is chromosome, extra chromosomal, circular, double stranded DNA which are capable of replicating independently. This plasmid carries in for antibiotic resistance, also carries in for uh, uh, resistance to heavy metal, and plasmid can be transferred from one bacteria to another bacteria and transfer resistance gene through conjugation, and this conjugation occurs through a component called sex pili. The hair-like projection that arise from the cell surface is called pili. Hair-like projection from the cell surface is called pili. It is composed of pilin protein. Pili is of two types. Ordinary pili and sex pili. Pili is two types, ordinary pili and sex pili. Ordinary pili mediates attachment to the cell surface, and sex pili participate in conjugation. Sex pili helps in conjugation. This whip-like structure that arises from the cytoplasmic membrane. Whip-like structure that arises from cytoplasmic membrane is called flagella. It is called flagella. It is composed of flagellin protein. Flagella is composed of flagellin protein. So difference between pili and flagella is pili arise from cell surface composed of pilin protein, and the function of pili is that mediates attachment to the host cell surface, and sex pili helps in conjugation. On the other hand, flagella arise from the cytoplasmic membrane and it is the organ of locomotion. That means this flagella helps in motility of the bacteria. It is composed of flagellin protein. And flagella is of four types. Number one, peritrichous flagella. This peritrichous flagella means flagella is distributed around the bacterial cell. Example, E. coli, Salmonella. E. coli Salmonella has peritrichous flagella. Number two is amphitrichous. Flagella. Amphitrichous flagella means there will be single flagella at each pole. That is called amphitrichous flagella. Example is Pseudomonas. This Pseudomonas bacteria has amphitrichous flagella. Number three is Lophotrichous flagella. Lophotrichous means there will be a tuft of flagella at one pole. A bunch of flagella at one pole. Example is Helicobacter pylori. This Helicobacter pylori has Lophotrichous flagella. And number four is monotrichous flagella. Mono means one. That means there is a single flagella at one pole. That is called monotrichous flagella. And example is Vibrio cholerae. This Vibrio cholerae bacteria has monotrichous flagella. This structure is called mesosome. This is ribosome. The function of ribosome is to synthesize protein. Bacterial ribosome is 70S and it is subdivided into 50S plus 30S. Bacteria doesn't have true nucleus and it doesn't have nucleolus or nuclear membrane. It is just a single molecule of coiled DNA. This is called nucleoid. This is called nucleoid.